Hello, welcome back to DNA Patshala. In every flower, there is a small system working silently to produce the next generation. Today, we will understand that system step by step. Just follow the flow once, and this topic will become very easy. Let's start from the basic unit. The male reproductive unit of flower is called stamen. It has two parts, filament and anther. Filament is just support. Actual work happens in the anther. Here, don't get confused. Male reproductive unit is stamen and not the anther. Okay? So, filament simply holds the anther in position. Think of it like stand holding a bulb. Light comes from the bulb, not the stand. Same way, function happens in anther. Anther is usually bilobed, meaning it has two lobes. Each lobe contains two pollen sacs. So, there become totally 4 pollen sacs. That's why it is called as tetrasporangiate. Okay, anther is bilobed and each lobe has 2 sacs. Therefore, there are total 4 pollen sacs. Remember it for your NEET and CT exam. This is very common exam trap, so don't confuse it. Now, if we look inside, each pollen sac is called a microsporangium. It has 4 layers and in the center we have sporogenous tissue. Let's quickly go layer by layer. First is epidermis. It is a outer layer. It has simple role, that is protection. Nothing complicated. Next is endothecium. It has fibrous thickenings and helps the anther to open and release the pollen, that is Anther Dehiscence. Important point, it is absent in aquatic plants. Remember, okay? Middle layer are temporary. They degenerate as the anther matures, so no major role. Innermost layer is tapetum and this is very important. It provides nutrition and materials needed for the pollen development. Without tapetum, proper pollen formation cannot happen. Okay? Tapetum is very important. It gives nutrition to the pollen grains. Don't forget it. At the center we have sporogenous tissue. These are diploid cells which form microspore mother cells, that is MMC. Now begins microsporogenesis, that is the formation of pollen grains. MMC is diploid and undergoes meiosis, so chromosome number reduces. As a result, we get haploid microspores. If meiosis is not clear, I have already made a separate detailed video on it. You can check that later or I will give you in the I button. Let's see the stages. First, MMC is formed. Then it undergoes meiosis in 2 steps. Finally, 4 haploid cells are produced. These 4 cells together form a microspore tetrad. Why tetrad? Let's see what is microspore tetrad. Tetrad simply means a group of 4 microspores temporarily attached. They are surrounded by a callose wall which is later dissolved by callase enzyme. After that, those 4 microspores become free. Now, each microspore develop into a pollen grain and pollen grain is called the male gametophyte. Understood? MMC undergoes meiosis to form 4 haploid cells. These haploid cells are called as microspore tetrad. These tetrads are simply held together by the callose wall which will dissolve later by the callase enzyme. And each microspore became free and each microspore will develop into a single pollen grain and this pollen grain is called as male gametophyte. Okay, now let's see the structure of pollen grain. Pollen grain has 2 layers, outer exine and inner intine. Both have different roles. First is exine. Exine is made up of sporolenin which is highly resistant. It protects the pollen from damage. It also has the thin regions called as germ pores. Why exine is needed? Why protection is needed for the pollen grains? Because they travel from flower to flower by the process called pollination. We will do another detailed video on pollination. Okay. Next is intine. Intine is the inner soft layer. It forms the pollen tube during the germination and pollen tube come out from the germ pores which is present on the exine. Okay, why germ pore is present on the exine? Because it helps to form the pollen tube during the germination. Let's see what is the cell content. Inside pollen grain there are 2 cells, vegetative cell and generative cell. Vegetative cell forms the pollen tube. So role of vegetative cell is formation of pollen tube and role of generative cell is to form the two male gametes. How will you remember it? Generative cell generates two male gametes. Okay, simple logic. One builds the path and one provides the gametes. The most pollen grain are released at 2-celled stage, but some plants like cereals releases them at 3-celled stage, that is after the formation of male gametes. Remember it for your NEET exams. Next, Anther Dehiscence. When pollen grains mature, anther dries and opens. This process is called as Anther Dehiscence. Which layer of anther is helping for anther dehiscence? Comment me down, okay? And pollen grains are released in the air. Before we end, let's quickly revise the full flow in one go. Stamen is the male reproductive unit. Anther is where pollen formation happens. Tapetum provides nutrition. Microsporogenesis involves meiosis, that is reductional division, and pollen grain is the male gametophyte. If this sequence is clear, most questions become straightforward. Now let's go through some quick NEET facts. These are direct question lines. Anther is tetrasporogenate. MMC is diploid. Microspores are haploid. Sporulanin forms exine and callase enzyme breaks the callose wall. These are easy marks if you remember them properly. So, if you look at the full flow, sporogenous tissue will form MMC. MMC will undergo meiosis. After meiosis, microspores are formed. Tetrads, remember it, okay? Then, formation of pollen grains. Okay, this entire process becomes very logical. And once the flow is clear, this topic is not about memorizing. It's all about understanding. So, thank you for watching. See you in the next video for the next process.